Wow, wasn't that incredible seeing a seed transform into a full-size radish plant? We don't realize it, but plants do grow and move. And these time-lapse videos show us how dynamic their growth really is. I love watching fascinating videos like this. Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. And today we'll continue my lecture series on plant physiology.
We will be discussing the fascinating topic of plant growth. Personally, this is the most exciting topic I will cover in my lecture series. I have spent most of my career trying to understand growth, how it happens, what are the limiting factors, and how can we modify it and improve it. Plant growth is so fundamental to our food supply, our industrial supplies, and beauty in our environment. Stick around as we do a deep dive into the structure and influence of cell walls, into cell expansion, and to what limits cell expansion and plant growth from the cell to the whole plant. As always, I like to acknowledge the great scientists who have significantly contributed to our understanding of the world around us. Today, we highlight three individuals who've had a profound impact on the study of cell expansion and plant growth. On the left, Roderick Hunt from the University of Sheffield. His work on plant growth analysis and mathematical equations has provided invaluable insights into plant growth processes. In the middle is John Boyer of the National Academy of Sciences, a pioneer in understanding water uptake and plant growth. Boyer also developed techniques to measure key physiological parameters. Daniel Cosgrove on the right from Penn State University is a leading researcher on cell walls and cell expansion, contributing extensively to our knowledge of plant growth mechanisms. All of these scientists are brilliant. Plant and crop science are so much more advanced because of their efforts and contributions. I personally have benefited from their knowledge and interactions and hope to share that with you in today's lecture. Plant growth depends on three key processes: cell division, cell expansion, and differentiation. This lecture primarily focuses on cell expansion and why it's so important for plant growth. We will also consider long-term growth of the plant and coordination of growth of different plant parts or organs. There are two distinct types of cell expansion. The first is tip growth. It occurs in specialized cells like root hairs and pollen tubes. These cells expand at the tip of the cell, which is called polar growth. It's dependent upon a large infusion of cell wall materials into the cell wall space, which involves cytoplasmic calcium and exocytosis. We will not go into this type of growth in this lecture. The other form of growth is called diffuse growth, and this is controlled in part by the properties of the cell wall. It's dependent upon the synthesis of cell wall materials and the orientation of the cell wall microfibrils. In this next part, we will go into this in some detail before we move on to the more comprehensive aspects of cell expansion and plant growth. But before we do that, let's just remember that all cells originate from meristematic cells, starting as small isodiametric generic cells before differentiating into specialized cell types. Each of these cells will have different sets of controls that make them into the cell type that they are. They could become a trichome, a xylem vessel, a pavement cell in the epidermis of the leaf, a guard cell, or even cortical cells in the root. And this lecture will concentrate on cortical cells, which make up the bulk of a plant's structure. Cell expansion begins with the cell wall. It starts with the sliding of cellulose fibers or microfibrils within the cell wall structure that loosen the cell wall and allows turgor pressure to extend those walls. The wall is composed of multiple layers of cellulose that are laid down as the cell develops and grows. The organization of these layers and the orientation of those cellulose microfibrils within become even more complex as the cell matures. The complexity of growth increases as we scale up from individual cells to tissues and organs. As I mentioned in the previous lecture on cellulose synthesis, the enzyme responsible for synthesizing cellulose is located in the plasma membrane. As we see here in this image with the CESA side view, CESA being CESA, an abbreviated name for cellulose synthase, we can see a chain of cellulose forming, which is a beta-1,4-glucan. On the cytoplasmic side, a glucose is being added to the end of that chain via a UDP glucose, allowing the cellulose chain to extend into the cell wall space. In the last lecture, I mentioned that the cellulose synthase complex was composed of hexamers of cellulose synthases. In that lecture, based on older data, each of these hexamers were thought to be composed of 6 cellulose synthases. However, recent updates indicate that it's more likely that there are 3 cellulose synthases, and thus 18 chains of cellulose are crystallized into a single microfibril. In the model below, we can see an artist's conception of the structure of the cell wall, with long chains of cellulose microfibrils intertwining with each other and interacting with each other. In certain spots, we can see xyloglucan, which is a hemicellulose that interacts with the cellulose microfibrils, and also pectins intertwining in between and within the fabric of the cell wall. On the right-hand side, we can see that the cellulose is organized in layers, showing how those layers form in different orientations over time on the exterior of the cell. If we expand that view from the cell wall to the whole cell view, then we can see the dynamics of multiple factors that are involved in cell wall expansion. On the left-hand side, we can see in part A, cell wall synthesis, which consists of the cellulose synthase complexes moving along cortical microtubules, which are linked to those cellulose synthases and guides them through the membrane. In the right-hand side B, wall assembly, we can see another component to cell wall synthesis that is going on simultaneously. This involves exocytosis of other cell wall materials, the matrix polysaccharides, which include xyloglucan and pectin that we just saw on the previous slide. These are synthesized in the Golgi apparatus and secretory vesicles before being secreted into the cell wall space. In addition, there are wall enzymes that are released in this process as well, out into the exterior apoplastic space. In C, the wall loosening section of the image at the bottom, we can see other factors that are important. There are proton pumps in the plasma membrane, which pump protons into the extracellular space, causing the apoplast to become acidic. This is important for growth because of a specific enzyme discovered by Daniel Cosgrove known as expansin, abbreviated here as EXPA or EXPA, which is sensitive to the acidification of the wall. These expansins actually loosen the cell wall and allow the cell wall to enlarge. It only enlarges if the wall is under stress. That is, if there is a force for extension, for example, turgor pressure. As you recall from my previous lectures, turgor pressure is generated by the movement of water into the cell, causing the cell volume to increase. The resistance of the cell wall causes an increase in turgor pressure, much like the resistance of a tire when air is pumped into it. Water is taken up from outside the plasma membrane because the water potential is lower inside the plasma membrane. This can occur because the solute potential is lowered, which can be a result of the uptake of sugars and potassium or other molecules and ions into the cell. Alternatively, the water potential can be lowered inside the cell by a decrease in cell turgor. This is what happens here when an expansin loosens the cell wall. There is a slight drop in turgor pressure. More water moves into the cell, causing a rise in turgor pressure, causing further increases in cell wall extension. And this cycle continues to repeat until one of these processes slows or stops. The most important point here is that this growth process starts with the loosening of the cell wall. In this figure, we can see how Daniel Cosgrove was able to identify expansin, the protein responsible for cell wall expansion. This was a brilliant piece of research. He used a stem section that was rapidly expanding in a specific area of the germinating seedling. He excised that tissue and extracted the proteins from the tissue cell walls into different fractions. He added each of the protein fractions back to a piece of growing section that had previously had its proteins inactivated by heat treatment. The growth or extension of this tissue was measured with an LVDT, which is a linear variable differential transformer, a device I will explain later in this lecture. The tissue is placed into an apparatus and a weight is hung on the device to apply a stretching force to the tissue. The cell walls begin to extend at this point, what he calls wall creep. The increase in length is captured and plotted in figure B on the right as the gray line or control. When adding different fractions of the extracted protein to this control tissue, he found one fraction in particular that increased the rate of wall creep. This cell wall protein was isolated and given the name expansin. You can see here that when expansin is specifically added to the inactivated tissue, that there's a rapid increase in cell wall extension or creep. He also found out later that if he added expansin to a piece of cellulose paper, it would cause the paper to loosen. Pretty nifty stuff. So how do expansins increase cell wall extension? In this figure here, we can see the concept that Daniel Cosgrove has of how these expansins work in a cell wall between the cellulose microfibrils. The red dots represent expansins in the cell wall. It turns out that these expansins do not appear to have enzymatic activity. That is, they don't seem to cut these fibers in the cell wall, but in ways that are not fully explained, appear to be able to wedge themselves between the cellulose microfibrils, helping them to pull apart. You remember that cellulose microfibrils hydrogen bond to each other. By wedging in between the cellulose microfibrils, it's thought that they can break these hydrogen bonds, reducing the resistance to slippage of these cellulose microfibrils in the wall. This makes it easier for the cell wall to expand. The orientation of the microfibrils can impact the direction of cell expansion and cell shape. This is illustrated here. A random orientation will allow the round shape or an isodiametric cell. However, when cellulose microfibrils are in a transverse orientation, then expansion can occur in only one direction, but not in the other. As you recall, the cellulose microfibrils are as strong as steel, and in this case act like the hoops of a barrel, restricting expansion in the lateral direction and allowing only expansion in the longitudinal direction. Thus, the cellulose microfibrils are shaping and directing the expansion of the cell wall. Cells don't grow forever. The walls can reach a limit in which they can no longer expand. It's unclear what all the controls are for this, but one hypothesis, known as the multi-net hypothesis, where the cell wall layers begin to limit the expansion by creating more resistance to expansion. This is due to the increased thickness of the cell wall and the orientation of the microfibrils. Remember that newly synthesized cellulose microfibrils are continually deposited on the inner surface of the wall. As a cell expands, the outer walls are stretched. This can cause a shift in their orientation. The transverse orientation allows for easy extension of the cell wall in one direction. However, as the orientation of the cellulose microfibrils in different layers shifts, the resistance begins to increase, resulting in a slowing of growth and eventually a cessation of growth. Okay, let's pause for a moment to review the factors that regulate cell expansion. These include the cell wall itself, the process of wall synthesis and loosening, and water uptake. Water uptake is crucial because even if the cell wall is loosened, the cell won't expand unless water increases the cell's volume. Without that increase, there won't be enough pressure to push the wall outward. Think of it like a syringe. If you block the opening with your finger and pull the plunger, without a needle, of course, you can't pull it back unless air or water moves in. Similarly, for a cell to expand, water must enter. For water to flow into the cell, the water potential inside must be lower than outside. This lower water potential is achieved by the uptake of solutes, which lowers the solute potential and in turn the water potential. If you're not familiar with these terms, I recommend revisiting my lecture on water uptake and transport. In summary, plants can regulate cell growth rates by controlling cell wall synthesis, wall loosening, water uptake, and solute uptake. These regulatory factors are captured in the Lockhart model for cell growth. First, we will talk about growth as a relative growth rate, abbreviated here as RGR. We will define this more clearly in a few more slides, but simply say that it's a proportionality of growth relative to the original size of the cell. This equation is beautiful. If you don't like equations, then I hope you will appreciate it after this lecture because they are very useful and allow you to visualize or analyze the regulators controlling growth and how they work with each other. So this first equation, RGR equals M times P minus Y in parentheses. This is a description of the relative growth rate based on pressure and the properties of the cell wall. M is known as the cell wall extensibility. This is a measurement of how fast the wall slides across itself once there's sufficient pressure to extend it. P is the trigger pressure of the cell, and the rate of growth is linearly dependent upon how much trigger pressure there is. If there's more trigger pressure, it will expand proportionately faster. However, the wall has some resistance to it, which we call the yield threshold of the wall. You can apply a little bit of pressure or force to the wall and it doesn't extend. However, once that minimum pressure is reached, the wall starts to yield. It starts to extend and then extends at the rate of the cell wall extensibility. So M and Y are two different properties of the cell wall. P minus Y is also known as the effective trigger, as it is the effective trigger that actually drives the growth rate. Relative growth rate can also be described using the principles of water uptake. A plant cell can only expand if it takes up water. That's why drought stops plants from growing. In part, it stops the plant cells from expanding. So we know that the relative growth rate can be described as equal to the hydraulic conductance, which, if you recall, is the inverse of resistance. It is a measure of the ease in which water can move into the cell. In this case, at the cell level, the barrier for water movement is largely dependent upon the plasma membrane, which is a huge resistance to water flow. It's also dependent upon a water potential gradient. That is the difference between the water potential inside and outside the cell. The greater the difference, the faster the rate of water flow into the cell. One minor note here is that hydraulic conductance is a term that has two components to it. It's dependent upon the area of the cell and the hydraulic conductivity LP. John Boyer further developed the Lockhart model by combining these two separate equations into what is known as the combined Lockhart equation. But before we get into that, let's look at these two equations in a graphical sense. In this figure, we have two lines. The solid red line represents a cell wall limiting equation. That growth rate is equal to m times psi p minus y, where psi p is the same as p for turgor pressure. And we have another red dashed line that represents the equation for water uptake, or growth, with a change in volume over a change in time equal to A for area times LP times the water potential gradient delta psi W. Remember that A times LP is equal to L. These are shown in relationship to turgor pressure on the x-axis. The rate of cell expansion on the left y-axis and the rate of water uptake on the right y-axis. If we look at this, we can see the demonstration of the yield threshold, where in this case, where the growth or rate of expansion doesn't increase until it reaches above 0.3 megapascals of turgor pressure. The slope of this line is m, the cell wall extensibility. On the dotted line, we have a decrease in pressure, allowing for more rapid growth because it increases the water potential gradient. The pressure begins to decline, followed by an increase in water uptake. Steady state growth is attained only at this point where the two lines intersect. Each line represents a growth rate controlled by the water uptake, or the cell wall rheology. Any imbalance between the water uptake and wall expansion will result in changes in growth rate. For example, if the cell wall loosens or yields at a faster rate, then turgor pressure will decline. This decline will cause a slowing of water uptake, and so expansion will fall back to the stable intersection. Putting these two concepts into one single equation, we can combine those two individual equations. If you don't believe me, I challenge you to solve it. I used to give this as an extra credit problem in class. Clue: there are several ways to find the solution to this problem. One way is to set up a solution with simultaneous equations. The combined Lockhart equation now is defined as relative growth rate, which RGR is equal to m, the cell wall extensibility, times L, the hydraulic conductance, divided by m plus L, times the water potential on the outside of the cell minus the solute potential on the inside of the cell. These latter two terms is sort of similar to the pressure, but represents a slight variation from the previous equation. These two terms, minus y, the yield threshold, provides us a term for effective turgor pressure, similar to the previous equation. The term ml divided by m plus L is known as the growth coefficient, which I will give examples of in a moment. This is a very important equation, and I'll show you how we can look at these different terms with scientific methods to understand what is limiting cell expansion. This is also a linear equation with the growth coefficient behaving like m, the cell wall extensibility, but also taking into consideration the influence of hydraulic conductance. Note that we now know there are water channels in the plasma membranes that are called aquaporins, so plants are capable of changing their water transport pathways. To measure some of these growth parameters, Daniel Cosgrove used the stress relaxation technique. He took excise growing stem segments from growing pea seedlings, which were incubated in solution before being placed into a humid chamber. He measured the turgor pressure of a cell in the stem segment with a pressure probe. This is plotted on the y-axis. And he measured the turgor pressure over time, which is plotted on the x-axis. You can see that the turgor pressure of the control cell, the blue line, was relatively stable at approximately 0.6 megapascals. This was because it was in contact with a drop of water, which allowed water uptake, maintaining turgor pressure. The turgor pressure of the black line for a stem segment not in contact with water declined with time, but this decline in pressure stops around 4 hours after the start of the experiment. This drop in pressure occurs because the cell walls are loosening causing a decline in pressure. Since there's no uptake in water, the pressure continues to decline until it reaches the yield threshold of the wall, at which point the cell wall can no longer extend. To further document that this technique worked, to measure the Y and P minus Y, he applied auxin, a plant growth hormone, to another segment before placing it in the humid chamber. In this case, represented by the red line, the cell grew faster, causing the trigger pressure to drop faster, reaching the yield threshold in only about an hour. An elegant set of experiments. A limitation of this method is that it only measures some of the growth parameters. In particular, it assumes that hydraulic conductance is not limiting, and this allows one then to assume the Lockhart equation can simplify to relative growth rate equals to m times p minus y. It also assumes that the growth parameters are constant and not changing in response to wounding and the other environmental conditions. Here is another technique for measuring some of the growth parameters of leaf expansion. I developed this technique and published it back in 1991. I don't want to go into all of the details of it, but if you're very interested in it, you can read this paper that's listed here. Basically, the technique works where I use a corn plant with an expanding third leaf. I attach a Teflon line that is not stretchable to the end of the leaf. Previously, I tried a nylon line, but found that it was stretchable, which made my measurements inaccurate. I extended the line over a pulley and down to a rod moving through an LVDT, that's a linear variable differential transformer, or basically an extensometer that can measure the length or movement of an object. Technically, it is an electromagnet, and as a metal rod moves through the core of the magnet, it produces a voltage signal that can be captured by a computer. This voltage signal is proportional to a length, so an LVDT can be used to measure the extension of an object, whether it's a piston in a car or the enlargement of a corn leaf. As you recall, a corn leaf has an intercalary meristem at the base where the cells are expanding and enlarging. The rest of the leaf cells are mature and not expanding, so it has a very well-defined growing zone and makes a very good model for measuring leaf growth of a grass. Adding weight to the rod can mimic an increase in trigger pressure, although in reality it is a linearly applied tension and not an increase in three-dimensional trigger pressure. The effect of adding weight, in this case 30 grams, to the LVDT core on the leaf length over time can be seen on the right. The weight was added at 10 minutes and removed at 57 minutes. The added weight causes the leaf to enlarge or grow upwards more rapidly, which translates into an increase in the steady state growth rate of the leaf. There's a very rapid extension of the leaf, which begins to taper off with time and reach a new rate after about 30 minutes. We can collect the data between 30 and 57 minutes as a steady state rate. If you remove the weight, you see a rapid decrease in length. That is measuring an elastic component of the cell walls. What we're doing here is measuring two different properties of the cell wall. There's a plastic component and an elastic component. Pulling the weight off at the end reveals the elastic component, sort of like a rubber band when you stretch something and it pops back. But if it's stretched far enough, not in the case of a rubber band, but a cell wall or plastic fabric, you can get a permanent extension of the material, a plastic extension. This is what's shown here when adding a weight. There is a rapid increase in length to both plastic and elastic stretching of the material, but underlying that is the actual natural extension and growth of the leaf. That is revealed in the linear steady-state growth rate between 30 and 57 minutes, after all of the plastic and elastic extensions are essentially complete. Again, if you want more details, read my paper. In Figure 1 on the left, you can see a proportional increase in growth rate as more weight is added to the rod of the LVDT system. This measurement system is extremely sensitive and can measure very small movements over very short periods of time. In this way, we can rapidly make changes to the growth rate and collect the data as seen here in Figure 2. We can capture the leaf elongation rate in its natural condition while growing the plant in a hydroponic system. The corn leaves are growing at a rate of 30 micrometers per minute. If I add salt to the hydroponic solution, this lowers the solute potential of the solution and therefore also the water potential of the solution, making it more difficult for the roots to absorb water. There's an immediate decrease in the growth rate of the leaf. Eventually, there is some recovery of the growth rate to a new, lower, steady-state growth rate after about 4 hours. But I'm not going to go into the details of what physiological changes are going on in the plant during the period of recovery. See if you can guess, and then read my paper to find out. The responses of the plant are proportional to the amount of salt added, with more salt inhibiting the steady-state growth rate to a greater extent. In Figure 8 on the right-hand side, you can see that the relative elongation rate of the leaf increases linearly with the amount of weight added to the rod, here plotted as an applied elongation force. Plastic and elastic extension also increase linearly. The slope of the plastic extension line can be converted into a measure of the cell wall extensibility. Now here is where this technique becomes interesting. In another paper I published in 2003, I compare the effects of salinity on 3 grass species differing in their salt tolerance. 2 varieties of maize, here listed as corn, 2 varieties of barley, and a wild barley. Wild barley is more salt tolerant than cultivated barley, which is more salt tolerant than maize. The question was, were there differences in the growth parameters of these different grass genotypes when exposed to different concentrations of salinity? In this graph, the steady-state rate or relative growth rate from the previous figure is plotted against the amount of applied elongation force. The slope of the line is equal to the growth coefficient, or ml divided by m plus l. And the x-intercept of this line represents the yield threshold of the cell wall, something I called the apparent yield threshold of the cell wall, since it's an indirect measurement. Using this technique, one can measure all the growth parameters with a few other measurements like the water potential and the solute potential, and determine which of the combined Lockhart equation parameters were limiting growth. The response to 4 different levels of salinity after 5 hours of treatment for all 3 species were different. There's a lot to discern from this figure, so let's just summarize the results from this experiment. The yield threshold was affected by most salt-tolerant treatments and most species, but not in all cases. The yield threshold of H. jubatum, or wild barley, was not affected by salinity. The increase in Y can be interpreted as a stiffening of the cell wall due to salinity. So salinity increased the yield threshold of the cell wall. This in turn reduces the effective trigger pressure of the cells to elongate. In barley, the yield threshold was increased and hydraulic conductance was reduced. In other words, it was harder to take up water. In wild barley, only the hydraulic conductance was reduced. Note there was no effect of salinity on the hydraulic conductance of maize. In all species, there was no effect of salinity on cell wall extensibility. Another interesting point is that the growth parameters of both cultivars of maize responded to salinity in the same manner, as did that of the 2 cultivars of barley. The take-home message of this experiment is that salinity inhibits growth of plants in the early phases of salt stress by affecting different growth parameters depending on the plant species. One needs to be cautious when generalizing about the effects of salinity, as it is clear that plants can respond differently. How do we measure growth? That question is more complicated than you would think, so let's pause and think about how growth is defined or can be measured. A basic definition of growth is an irreversible increase in plant size, in volume, or in length. Growth can also be measured by counting the number of leaves, or measuring an increase in dry weight, or even fresh weight, or it could be defined as the increase in the number of cells. From an agronomic point of view, growth can be measured as an increase in yield, or the harvest index of a plant, HI, where the harvestable dry matter is divided by the total dry matter. For example, you could have corn kernels from a corn plant, and you would measure that weight divided by the total plant dry matter. As we will see, which particular choice you make can affect your outcomes and conclusions. For example, let's take a look at seed germination. We can see over time that the fresh weight, the yellow line, increases with time, but the actual dry weight, the magenta line, of the same seed decreases with time. This is due to seed respiration, using some of the carbon reserves of the seed, decreasing its weight. So the choice of growth measurement is important. This is a classic graph of growth in biology. This would be a growth curve for yeast or bacteria, for example, but it also applies to plants. In the early stages of growth, we have what is called a lag phase, which quickly moves into a logarithmic phase and begins then to slow down and eventually reach a stationary phase, or into a senescence stage, where there's actually a decline at the very end of the cycle. This growth curve can be defined mathematically. Knowing this equation that can describe this curve allows us to make real insights into how plants are growing and what processes might limit their growth. Let's dive into this a little deeper. In plant growth analysis, data are usually obtained from successive destructive harvests performed within the plant growth cycle, from which the growth rates are calculated. Two main approaches have been used toward estimating growth rates. In the classical approach, mean values of growth rates are calculated by formulae previously derived using data of 2 consecutive harvests, whereas in the functional approach, mathematical functions are fitted throughout the growth data over time, their differentiation providing instantaneous values of growth rates. Let's start here with a definition of the absolute growth rate. Growth rate is equal to the derivative of weight, W, which is dry weight, divided by the derivative of time, T. This is equal to the change in weight, shown here with a triangle, divided by the change in T, which is equal to the weight at time point 2 minus the weight at time point 1, showed here by the subscript 2 and 1, divided by the time at time point 2 minus the time at time point 1. This is typically expressed in days, but it's not limited to that. You could use hours or minutes if you could detect differences. You could use fresh weight or length instead of dry weight. Now, it turns out that the absolute rate is dependent upon the initial size of the plant and can be misleading at times, so that there is a better expression called the relative growth rate, which takes into account the original size of the plant. The relative growth rate, RGR, is equal to the derivative of weight, W, divided by weight, times the derivative of time, which is equal to the change in weight divided by the change in times 1 over W. This is equal to the expression of the natural log of W2 minus the natural log of W1 divided by T2 minus T1. In plant growth analysis, you can take this one step further. You can define relative growth rate as equal to NAR times LAR, where NAR equals the net assimilation rate, and LAR is equal to the leaf area ratio. We can define the net assimilation rate as the derivative of weight divided by the leaf area times the derivative of time, which is equal to the change of weight divided by the change in time times 1 over the leaf area. And the leaf area ratio is equal to the leaf area divided by the weight of the plant. These are very basic definitions and are very useful to understanding plant growth. Basically, we can call the net assimilation rate equivalent to the photosynthetic rate per unit of leaf area of the plant. And the leaf area ratio is a measure of the leafiness of the plant. How much leaf area does it have to capture light? These two variables are important for understanding the relative growth rate and the limitation to plant growth. I'll give you an example in a moment. To get a more in-depth understanding of this, I highly recommend you read the book by Roderick Hunt called Plant Growth Analysis that was published in 1978. This type of analysis is quite often used by agronomists to understand crop growth. Here's a graph of the growth of a hypothetical plant over time based upon the natural growth curve. Here, in the light blue dotted line and labeled with a W, we can see the lag and logarithmic phases, and then a stationary or senescence phase. If we took the derivative or broke up that blue line into a series of weight measurements and calculated the weight difference at different time points, using the formula for relative growth rate that I gave you previously, the red line would represent the plot for that relative growth rate. From it, you can see that in the early stages of growth, the relative growth rate is at its highest. And then as the plant grows into the logarithmic phase, relative growth rate begins to decline, ultimately reaching zero at the very end of the cycle. We can also derive the net assimilation rate and leaf area ratio. We can see the net assimilation rate in the black dotted line labeled as NAR. It's relatively linear for a while through about the midpoint, then starts to decline with time, whereas with the leaf area ratio, the increase in the leaf area ratio increases over time. What we can also observe is that the net assimilation rate contributes more to growth in the early stages of growth, and the leaf area ratio contributes more to the relative growth rate towards the end of the plant's life cycle. Let's look at an example of some of my own research. Using these techniques to understand how salinity impacted the long-term growth of corn. This is based upon a paper I published in 1994 in the Australian Journal of Plant Physiology. First, let's look at the figure in the upper left-hand corner. Some short-term measurements were made of a corn plant growing in a hydroponic system that I have described before, where we measured the growth rate or leaf elongation rate with an LVDT, shown here as the triangles. Simultaneously, we measured the photosynthetic rate of the third leaf with a photosynthesis meter. This is represented by circles. You can see in this experiment, similar to the previous experiments I showed you, that when you add salt at 30 minutes, there's a rapid decrease in the leaf elongation rate, which goes through a recovery phase, but only recovers to about 80% of the original leaf elongation rate. Now this is interesting. When you remove the salt, by changing out the solution with a fresh control solution without the salt, one gets a very steep increase in the growth rate above that of the original leaf elongation rate, which then declines and drops down, but eventually returns back to 100% of the original leaf elongation rate before salt treatment. So there was no permanent salt toxicity in this treatment. I'm not going to go into all the details of why physiologically that happens. The main point of this is to show you the fact that leaf elongation rate was altered and was independent of the photosynthetic rate. Clue, it is due to water relations in the leaf elongation zone of the leaf and the ability of the plant to absorb water. In a separate set of experiments, which are a bit complicated, there were two different corn varieties used, one more salt tolerant than the other, and there were two different kinds of treatments based upon the kind of salt that was added to the solution. But the solutions were approximately the same osmotic strength. It's not important here to discuss the difference, but one salt solution is more toxic than the other. One treatment is listed as sodium and the other one is listed as sodium plus calcium. The plants were grown over a period of 4 weeks or 28 days in the solutions. In the upper left-hand corner, we can see the total dry weight increase of the two plants. You can see the weights of the two control solutions, one for cultivar 3578 and the other for cultivar 3772. There's not much difference between the two cultivars. They are approximately the same. However, 3772 did have a higher dry weight than 3578 at the end of the experiment. These are represented by the circle symbols. The next treatment with squares is the sodium plus calcium treatment, which are much lower in dry weight by day 28 than control plants. And the triangles representing the sodium chloride treatment, the dry weights were even lower. A different way to look at this is to express the data as percent of controls, as in the figure B below. You can see that growth by day 28 was lower by about 20% of the controls for the sodium chloride treatment, but the addition of calcium increased the growth to a total dry weight of about 60% of the controls. But there were also immediate effects early on in the experiment, which were not easy to see in the figures expressing the total dry weight. The differences don't look significant because they're compressed so much in the figure. Okay, that gives us the basis of the treatments. Now let's analyze it with the plant growth analysis. We can plot the relative growth rate and we can see that in 3578, there's actually a little bit of an increase in the relative growth rate. However, the error bars displayed are overlapping. This means that they're relatively the same and there are no significant statistical differences between them. But if the error bars are apart from each other and are not overlapping, then there are some significant statistical differences between the data points. Basically, among those treatments, the relative growth rate was impacted, but not to a huge extent in the cultivar 3578. In 3772, there was a very distinct difference early on in the beginning with salt treatments, and then later on, particularly in the sodium chloride treatment, but not in the sodium plus calcium treatment. What does this mean? We need to look at the next two figures, which are the net assimilation rate and the leaf area ratio, to understand this. If we look at the net assimilation rate figures, we see that all the error bars are overlapping and they're relatively flat, meaning that the net assimilation rate was unaffected by the salt treatment, similar to the photosynthetic measurements that were done in the short-term experiments, and confirming that the changes in the relative growth were largely due to the impact on the leaf area ratio, or the leafiness of the plant. This type of analysis reveals an important insight into how salinity is having a long-term impact on the growth of corn plants. It is the inhibition of leaf expansion that is limiting the growth of the plant, not the photosynthetic rate. This reduces the plant's ability to capture the light energy from the sun. The overall photosynthetic apparatus is functioning and not limiting growth. We will discuss the salt tolerance of plants in more detail in a future lecture. Now, as I said earlier, the absolute growth rate is not a good measure of growth. Here's an example of why that is. Let's suppose we have two plants. Plant number 1 is equal to a starting weight of 10 milligrams, and plant number 2 starts out at 12 milligrams. That's only a 2 milligram difference, a very small amount. Now let's also suppose that these two plants grow equally fast at a relative growth rate of 0.25. Let's compare the two plants again 21 days later. Plant number 1 would now be equal to the size of 1,906 milligrams. Plant number 2 would be equal to 2,287 milligrams. We have a difference between the two plants now of 381 milligrams. That seems like a huge difference compared to 2 milligrams, and we'd say that plant number 2 grew so much faster than plant number 1. In fact, we would be fooled because we now know that they grew at exactly the same relative growth rate. This is why if you were a scientist studying the growth of a plant and you just did a single harvest with some sort of treatment effect that you're studying, and you came out with two different sized plants that were very different, you would be fooled into thinking that they had grown much more in one treatment than in the other. But it was simply due to the fact that the initial sizes of the plants were different in the beginning. I learned this the hard way with my very first experiment on plant growth and realized after doing an experiment with a single harvest that it was very important to actually do multiple harvests to get a more accurate measure of my treatment effects. Let's look at another example where the importance of time can have an important effect. For example, let's say we have a difference in seedling germination after planting. In this example, we have 3 different plants, all of which are equal in size. Plant number 1 is equal to 10 milligrams. Plant number 2 germinates 2 days after plant number 1. And plant number 3 germinates 7 days after plant number 1. Again, all plants grow at the same relative growth rate, which was equal to 0.25. As in the previous example, if we look at their growth 21 days later, plant number 1 is by far the largest plant, weighing 10,966 milligrams, versus plant 2, which is equal to 6,651 milligrams, versus plant 3, which germinates 7 days later, is equal to 1,906 milligrams. So again, if you're in the field examining plant size in the field, one could easily draw the wrong conclusions based on the plant size, as if they were growing at different rates, when in fact they just germinated at different times. The plant growth analysis is very important at helping you interpret your results more clearly and helps us prevent drawing the wrong conclusions from our data. Measuring plant growth is a little trickier than it looks. There are other ways that one can measure growth with the new technologies that are coming out, both at the cellular level and at the whole plant level. If we look at the left figure with 3D confocal microscopy, one can actually image a root and all the individual cells. You can look at each and every cell, how it's growing and elongating within that root tip. One can also view whole plant growth using 3D laser scanning to try to capture the growth of the entire canopy of a plant. These are just 2 examples. There are other approaches as well, and new ones being developed all the time. Here's an example of where scientists are looking at growth and they show that roots actually grow towards water. This is a phenomenon known as hydrotropism. One more thought before we conclude this lecture. How does a plant divide up its resources and coordinate the growth among its organs? How does it know how to control how much roots to grow versus how many leaves to grow versus how many fruits to bear on the plant? Let's consider some factors that are involved. In this consideration, we will assume that there are abundant resources of light, water, and nutrients, that we are in optimal growth conditions where there are no resource limitations. This links us back to our discussions of sugar transport in plants in previous lectures. Organs compete for sugar through what we defined as source-to-sink relationships, and roughly follows the law of supply and demand. The source is the production of sugar by photosynthesis. The demand is the number and strength of the sinks. For example, the roots and other non-photosynthetic organs, such as tubers and fruits, they would be sinks. High demand will drive the activity of photosynthesis. The higher the demand, the greater the rate of photosynthesis. The reverse is also true. If you reduce the number of sinks, like removing fruits from a plant, you'll reduce the photosynthetic rate. Go back to my lectures on photosynthetic regulation to get a glimpse of what I'm talking about here. Photosynthesis, sugar metabolism, and sugar transport are subject to feedback regulation. Attempts at increasing the rates of photosynthesis of plants have not succeeded in increasing productivity due to this complex feedback regulation of the high concentration of sugars on the photosynthetic rate. The important point here is that it is the sink demand that controls photosynthesis and growth. Ultimately, the size and number of sinks are controlled by the plant's DNA, its genetic blueprint. Let's give an example here with a grapevine. On the left is a figure of some structural and anatomy terms for you to understand. Grapevines have roots, a trunk, some large branches or arms called cordons, and they have shoots coming off of the cordons, which produce fruit. Grapevines are perennials that reproduce over many years. In the right-hand figure, let's look at the growth of these different parts of the plants over the course of a season. We'll see that demand changes within the season, that there are a large number of sinks, and these sinks are all competing with each other at different strengths at different times of the season. So on the left-hand y-axis, we have shoot elongation rate and also the activity of the growing root tips. On the right-hand y-axis, we have the rate of change in the trunk radius and the rate of berry growth. Okay, so let's start on the left-hand side at day 0 when bud break is occurring. If we follow the growth of roots, we see that there is a solid line with a few dashes in it, and we see that root growth increases at bud break and peaks about 50 days later at anthesis. Anthesis is when flowering is occurring. They represent a large set of sinks that are developing. At that point then, root growth declines rapidly to mid-season by about day 140 and then increases again at the end of the season. Now let's look at shoot growth. This is the medium-sized dash line. We can see that it's very high at the beginning of the season, and then it declines and comes to a complete stop at around day 110. It starts off very rapidly and declines. Shoot growth initially after bud break is very dependent upon the stored starch in the trunks and roots from the previous seasons. The growth rate of the shoot elongation rate declines with time, but the shoots are still there doing their photosynthesis. They just aren't growing and extending very much. Now let's look at the most important thing, which are the berry clusters. The berries start their development at just after anthesis. We see a very large increase in the berry growth here as a solid line, which increases at about a maximum at day 80. And then its growth rate slows for a period of time before it has a second increase in growth rate that peaks again at about 120 days. After that, the growth rate continues to decline consistently throughout the rest of the season. Note that at berry harvest, the root growth starts to increase. The sink demands of the berries have been removed. Now let's look at the final competing sink here, which is the trunk. The trunk is displayed here as a very short dotted line. We can see that the trunk growth starts at about day 10, growing more rapidly and peaking just shortly after anthesis. As the berries start to grow, they compete for the sugar resources and the root, shoot, and trunk growth rates decline. Trunk growth declines gradually until we get to about day 120, where the berry growth rates begin to slow down. We see another increase in the growth rate of the trunk that peaks about day 140. This figure shows the competition between different sinks, how one sink can influence the growth of the other sinks. This, in large part, is driven by the berry growth, which is the strongest and probably the most important sink for the plant. Again, just to remind you that it is the leaves that are producing the sugars, which are loading sugars into the phloem, driving up the pressure in the phloem to a higher pressure. The sinks, in this case, are removing the sugars at the other end of the piping system, the phloem piping system, which reduces the turgor pressure at that end. These actions create a pressure gradient, and so the sugars flow in the direction of the sinks due to mass flow down its pressure gradient. It is the sinks that control the removal of sugars, that directs the sugars in one direction or another. As the berries are removing sugars from the phloem, more sugars naturally flow down the pressure gradient to the berries rather than to competing organs such as the roots or the trunk. In this way, growth is coordinated by the competition. The greater the pressure gradient, the path with the greatest pressure gradient is going to have the most flow. Of course, this is a rather simple statement, and things are always more complicated than they seem at first. There are plant hormones that we will talk about in future lectures, which can affect plant growth and sink strength. I'm going to end this video with a few clips of tropic growth responses that plants have that are influenced by light and some of these growth regulators. I know you'll enjoy them.
But before we do that, let's summarize this lecture. We can state now that plant growth is dependent on an increase in size or mass, that water and carbon accumulation are key components driving cell expansion and the capture of light energy, that sink demand drives photosynthesis and growth, that scientists have developed very sensitive ways to measure growth and determine the limiting factors restricting a plant's growth. Now let's finish with a few short videos of tropic growth responses captured by plant physiologist Dr. Roger Hangarter from Indiana University.
Well, that's it. I hope you enjoyed this video. If you like this video, then please like it on my YouTube channel. And consider subscribing to my YouTube channel where you'll be notified of other interesting videos that are coming out in the future. There'll be other plant physiology lectures, and I also have videos on viticulture and winemaking or enology. So have a great day.